Bismillahir Rahmanir Rahim. Welcome again to Derma Immunology. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University. Our video today will be about the antimicrobial peptide LL37 generation and structure. As we all know, the innate immune system serves as a protective barrier against a wide range of pathogens and frequently triggers a series of inflammatory reactions. The innate immune molecules are synthesized by epithelial cells or lymphocytes, and while there are various antimicrobial peptides, the Cathelicidin LL37, referred to as LL37 peptide, is the only member of the family of human cathelicidins. LL37, which is a cathelicidin antimicrobial peptide, is produced by epithelial cells, neutrophils, and lymphocytes, and play a key role in protecting eukaryotic organisms against bacterial, viral, and fungal infections. And in addition to its antimicrobial characteristics, LL37 serves as an immunomodulator, potentially contributing to the onset of autoimmune diseases. So another non-antimicrobial role of this cathelicidin involves its capacity to create complexes with DNA, which can trigger plasmacytoid dendritic cells to generate type 1 interferon, thus affecting the progression of autoimmune conditions like systemic lupus erythematosus. The chronic activation of plasmacytoid dendritic cells through LL37 DNA anti-DNA complexes recognized by receptors on plasmacytoid dendritic cells initiates endocytosis and subsequent toll-like receptor 9 recognition. This process leads to plasmacytoid dendritic cells activation and increased type 1 interferon production, which is a critical factor in early autoimmunity development in systemic lupus erythematosus patients. LL37 was originally believed to be exclusively found in the secondary granules of neutrophils. However, it's now understood that this molecule is produced in various cell types, including natural killer lymphocytes, macrophages, and epithelial cells found in the intestine, airway, genitalia, eye surface, skin, and certain endocrine glands, among others. The inherent presence of LL37 in various epithelial cells highlights a significant role in protecting against disease caused by pathogens. It's well established that LL37 levels increase in reaction to factors like wounds, ultraviolet radiation, damage to the epithelial barrier, specific components of bacterial cell walls, and endoplasmic reticulum stress, among numerous others. Two significant signaling pathways contribute to LL37 production. Vitamin D induces LL37 expression, which occurs naturally in non-inflammatory conditions, and the second pathway is through nuclear factor kappa B induced expression, which is activated during periods of inflammation and endoplasmic reticulum stress. NF kappa B affects not only the expression of the protein but also its release from cells and its activation through proteolytic mechanisms. So LL37's goal is to promote the restoration of epithelial functions following an immune attack. LL37 exists as a precursor molecule stored in granules within neutrophils, natural killer cells, and mastocytes. It is released in response to infections or tissue damage triggered by toll-like receptors or cytokine signaling. Initially, the inactive precursor human cathelicidin antimicrobial peptide 18 is liberated into the extracellular space where it undergoes cleavage in its C-terminal domain. This cleavage process is carried out by serine proteases from kallikrein family in keratinocytes and by proteinase 3 in neutrophils. The neutrophils, due to the high concentration of LL37 they release at sites of inflammation, play an important role as they amplify the immune response to the point of eradicating the infection. LL37, a proteolytic component of cathelicidin, serves as a defensin by integrating into membranes of bacterial pathogens. It also acts as an alarmine, stimulating chemotaxis of innate immune cells and affecting the structure and effectiveness of various cytokines. And just as a reminder, defensin is any of a class of peptides that are found in neutrophils and have antimicrobial and cytotoxic properties, while alarmines are endogenous molecules that are constitutively available and released upon tissue damage and activate the immune system. The direct impact of LL37 on neutrophils and the release of extracellular traps nets have been recognized as essential components of the immune defense against pathogens. However, nets are also significant factors in the development of chronic diseases and tissue-related pathologies. The antimicrobial peptides are parts of the innate immune system which serve as the initial host defense molecules. And there are more than 2,000 of these peptides, usually consisting of fewer than 100 amino acids residues, and they have cationic means a positively charged ion, and amphipathic is a molecule, especially protein, having both hydrophilic and hydrophobic parts characteristics. So these antimicrobial peptides are cationic and amphipathic. Many of the peptides are produced by proteolytic processing from a larger precursor protein. In humans and higher vertebrates, dozens of different proteases distribute into different structural classes and balance with equally diverse protease inhibitors. The result is a continually changing landscape of precursor proteins and proteolytic fragments. Various classes of antimicrobial peptides exist, with one prominent example being the human cathelicidin family, where human cathelicidin antimicrobial peptide 18 represents the exclusive member in humans. This 16 kilodalton cationic antimicrobial protein features a conserved N-terminal cathelic domain and an LL37 as a C-terminal antimicrobial domain, which can be liberated from the precursor protein through proteolytic cleavage, as I will explain in the next figure. In this diagram, we can see the precursor protein for LL37, which is the human cathelicidin antimicrobial peptide 18. And we can see here the C-terminal with the antimicrobial part LL37 that is cleaved by proteinase 3 in neutrophils, and maybe members of kallikrins like kallikrin 5 and 7 in epithelial cells. So we can get the antimicrobial peptide, the active part, which is LL37. LL37 and its mouse counterpart, mouse cathelicidin-related antimicrobial peptide, show a wide-ranging antimicrobial effectiveness. Also, LL37 has gained significant recognition for its involvement in the onset of inflammatory and autoimmune conditions like systemic lupus erythematosus, rheumatoid arthritis, psoriasis, atopic dermatitis, and atherosclerosis. And as I said before, LL37 can be found in the epithelial cells of the testis, skin, gastrointestinal tract, and respiratory tract. It's also present in various leukocytes, including monocytes, neutrophils, T cells, natural killer cells, and B cells. And as I said, LL37 originates from the human cathelicidin antimicrobial peptide 18 precursor protein through proteolytic processing by serine proteases, especially calicrans 5 and 7, and which are present in the epidermis, and the protease 3 primarily found in neutrophils. During this processing, it results in the formation of signal peptide, 30 amino acids, the catheliin, 103 amino acids, and the C terminal domain, the LL37, which is 37 amino acids, which becomes active following maturation. Peptides with reduced length, like what's called KR12, which lack certain amino acids from LL37, retain its activity. Also, some analogs, such as KR12-3, where specific amino acids are substituted or non-cleavable sequences, show heightened activity in comparison to LL37. LL37 is a peptide comprising 37 amino acids with 2 leucine residues located at its N-terminal. At a neutral pH, it has a net charge of +6. You remember that I said it's cationic and amphipathic. LL37 amphiphilic nature is related to the arrangement of 16 charged residues and the presence of hydrophilic and lipophilic functional groups within the alpha helix structure. Alpha helix structure is a secondary structure. Actually, alpha helix is a right-handed coil of amino acid residues on a polypeptide chain, typically ranging between 4 and 40 residues. In this photo, we can see the alpha helix structure of LL37. This unique composition explains the dual affinity of LL37 for both water and oil, hydrophilic and hydrophobic targets, and explains its multiplicity of functions. Also, the structural characteristics of LL37 in both 2 and 3 dimensions have been resolved in various conditions. This includes investigations involving full-length and shortened peptides, monomeric and oligomeric states, such as dimers, tetramers, and fiber-like structures, as well as peptide complexes with various ligands in diverse environments that include different detergents, lipids, and cofactors. These studies have shown a diversity of structures that has emerged from 2 dimensions and 3 dimensions of LL37, highlighting the remarkable flexibility of LL37 that adapts easily to the interactants it meets. When exposed to a phospholipid bilayer, LL37 tends to take an alpha helical conformation covering from residues 2 to 31, while residues 33 to 37 retain mobility. In water at micromolar concentrations, LL37 presents a distorted structure characterized by what's called circular dichroism, which is the property of an optically active medium of unequal absorption of right and left plane polarized light, so that the emergent light is elliptically polarized. In a summary, it means that LL37 has a great flexibility and changing its shape depending on the interactants with it or the medium. So the tertiary structure of LL37 provides the peptide with a diverse group of specific and biologically important interactions, ranging from the potential to inhibit viral infection to interesting applications in chronic neurodegenerative disorders. Recent examples involve the precise and strong binding between LL37 and the spike protein S1 of severe acute respiratory syndrome, SARS-CoV-2. This interaction prevents host cell binding by obstructing the receptor binding domain of S1. To demonstrate the adaptability of LL37, another binding region of the peptide engages with the ligand binding domain of angiotensin converting enzyme 2, which serves as the receptor for the entry of SARS-CoV-2 virus. On the other side, LL37 suppresses the aggregation of alpha-synuclein, a key factor in the progression of neurodegeneration in Parkinson's disease. So the anti-aggregation potency of LL37 prevents oligomer-induced cellular toxicity and may thus offer a promising therapy for Parkinsonian disease. My sources of information in this video include the first one is this paper, LL37, a multifaceted amphipathic peptide involved in netosis, which was published in Cells in 2022 August. My second reference is this paper, Cathelicidin, LL37, a new important molecule in the pathophysiology of systemic lupus erythematosus, that was published in Journal of Translational Autoimmunity in 2020. I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.